Assalamu alaikum everyone, my name is Rula Ismaiel and I'll be giving today's POW. We're going to be talking about the nitrogen disposal, and there are three main concepts that you need to know about. You have the enzymes involved, their cofactors, their substrates, their products. You also have the ammonia transport cycles, and you have two clinicals that you need to know. Now, for some background information, you need to know that amino acids are obtained from diet, which are called essential amino acids, de novo, which means they're made in the body, or through protein degradation, protein breakdown in case something goes wrong. In case the protein is wrong. Body proteins being degraded and resynthesized is called protein turnover. And yeah. Now, you need to know a concept called nitrogen balance. In a healthy adult, everyone in a healthy adult, they're in normal nitrogen balance. However, there are cases where there is positive nitrogen balance or there is negative nitrogen balance. Positive nitrogen balance means that there is more storage than there is excretion. This happens due to tissue growth or recovery from illness. Tissue growth basically means like a child that's growing up or in pregnancy. Now, negative nitrogen balance means that there is more excretion than there is storage, or there is less intake of nitrogen in the form of protein. And this occurs due to stress, trauma, burns, or illness, or malnutrition. Now, hyperammonemia can cause cerebral edema, convulsions, coma, and death. Hyperammonemia means an increase in ammonia in the blood. Okay, cerebral edema is increase of fluid in the cranial cavity, which puts a lot of pressure on the brain, causing seizures. Okay, which is convulsions. The urea cycle accounts for 80% of the excreted nitrogen, and 3% is eliminated from the kidney as ammonia. Now we're going to start off with amino acid catabolism, which basically there are two main steps to it. There's removal of alpha amino groups through transamination, glutamate, or oxidative deamination, which is straight up removal. Alpha amino group keeps the amino acid from oxidative breakdown. Now, the reason for this is you don't want the body, okay, to break down. You don't want the body to break down amino acids immediately. You want the body to use the amino acid for protein. Okay, so the alpha amino group protects that amino acid from breaking down. Now, formation of ammonia and alpha keto acid. You have your amino acid, we've done a bunch of stuff to it, then it reaches the liver so that ammonia can be released, so that it goes to the kidneys and, urea cycle. And then you have the alpha keto acid, okay, which is the carbon skeleton of amino acids, okay? Now, the carbon skeleton can be used as an intermediate in a lot of the energy-producing pathways, think MOL1 content, which later on produces carbon dioxide, water, glucose, fatty acid, ketone bodies. Ammonia mainly becomes urea, which gets excreted as urine. Now, this is a schematic, what do you call it? This is a schematic drawing of what's happening, basically, like a very rough summary. Just gonna turn on the pen. Okay, now the alpha amino group, which is this guy, the one that's causing all these problems, of alpha amino via aminotransferases, a transferase enzyme, becomes alpha ketoglutarate, which later on forms glutamate. Okay, these are big words, okay, but the main context of this, what it basically means is that this amino group, okay, can be put here through an aminotransferase. And you can see this being happening here. Like it got added to this molecule, okay? Now glutamate via oxidative deamination releases its ammonium ion, okay? Again, same thing, alpha-ketoglutarate and amino acid glutamate ammonium. Now you can see two enzymes that you need to know that are aminotransferases, okay? You have AST, okay, there we go, AST and ALT, okay? AST, aspartate aminotransferase with the help of, AST catalyzes the reaction between aspartate and alpha-ketoglutarate to give off oxaloacetate and glutamate, okay? So the enzyme would be here, okay? Keep in mind these reactions are reversible, okay? Aspartate, alpha-ketoglutarate, oxaloacetate, glutamate. However, aspartate tends to be in the opposite direction, so it's more lenient to go to this side. It goes the other side rather than to the side of glutamate, all right? Alanine, on the other hand, alanine aminotransferase, it catalyzes the reaction of alanine plus alpha-ketoglutarate, which gives you pyruvate and glutamate, okay? Please keep in mind the different energy intermediates, energy cycle intermediates, okay? So AST gives you oxaloacetate, ALT gives you pyruvate. We're gonna go into depth with this reaction in a second, okay? Transaminases, they're specific proteins. This is the first step, okay? They're specific proteins and they always result in glutamate. Glutamate is a temporary storage of nitrogen. That nitrogen can be used for amino acid biosynthesis, so the de novo, basically, okay? The de novo process. Now, there is a very important cofactor that's needed, which is pyridoxal phosphate, which is PLP, which is derivative of vitamin B6/is B6. Okay, now you can see how aminotransferase and PLP, they work together. So I have two of these, they become friends, and then I have a keto acid and a glutamate, okay? ALT functions towards glutamate synthesis. AST, on the other hand, the amino groups from glutamate goes to oxalate, which forms aspartate, which is a nitrogen source in the urea cycle, okay? AST and ALT are elevated in liver disease. Other aminotransferases can be elevated due to other problems like myocardial infarction, which is heart attack. Now, oxidative deamination occurs with a specific enzyme called glutamate dehydrogenase. Now, dehydrogenase means that a hydrogen is being taken away, okay? So I have glutamate deamination. So deamination means removing the amino group. So this, okay, to this, I'm removing that amino group to give me an alpha-ketoglutarate. Keep in mind that there are, There are cofactors that are used here. In transaminases, we needed PLP, and glutamate dehydrogenase, we need both NAD plus and NADP plus. Okay, it's the only enzyme that needs both / has specificity for both. Okay, if it's going to the direction of alpha-ketoglutarate, then NAD plus and gives you NADH and ammonia. So ammonia is being released from the glutamate. However, if we're going in the direction of glutamate, okay, the ammonia gets incorporated with the help of NADPH, okay, giving you NAD plus to give me glutamate, okay? You can see, like, to get technical about this, okay, you can see that this plus, okay, which is a hydrogen ion, okay, gets incorporated into the NAD plus to give me an NADH. Okay, so two pluses gives me an NADH. However, in this, okay, NADPH, okay, loses one of its pluses, if that makes sense, to give it to the ammonia so that it gets incorporated here. Okay, this is all chemistry talk. If you like chemistry or dislike chemistry, You don't really need it. I'm just trying to make sense about these cofactors. So please keep in mind in which direction it's like in which direction is the reaction going, and what in cofactor is being used. Now, during a high protein meal or low energy, there's degradation of amino acid. In meals, the excess amino acids are being degraded. However, in low energy, your own amino acids, the amino acid pool is being degraded for energy from the carbon skeleton. Okay, so the alpha ketoglutarate can become pyruvate or oxaloacetate, and that's all being used in glycolysis or gluconeogenesis. Okay, and other cycles. Transport of ammonia from muscles. Okay, this is one of the cycles that you need to know, but the bulk of it is that ammonia gets incorporated into glutamate. Okay, so it can carry it. Okay. Glutamate, however, can't go around in the blood as is. Okay, it needs to be contained. Okay, so to transfer the ammonia from the muscle to the liver, we use an amino acid called alanine. Okay, so glutamate with the help of pyruvate, okay, they come together, giving you alanine and alpha ketoglutarate. What is this reaction catalyzed by? Alanine aminotransferase. Okay, alanine moves into the liver, gets broken down to pyruvate, which is used for gluconeogenesis, glycolysis, whatever it may be. Okay, alanine broken down to pyruvate. Okay, alpha ketoglutarate with the addition of ammonia becomes glutamate. Okay, glutamate then stuff happen to it, becomes aspartate. Then you have ammonia, which results in urea. Okay, so keep in mind these are both ALT, alanine aminotransferases. Okay. The glucose-alanine cycle eliminate nitrogen and replenishes energy at the same time. So both things are happening. The amino group and pyruvate gives you alanine. The amino group is the ammonia on the glutamate, in the glutamate. The alanine from muscle goes to the liver. In the liver, amino group from alanine to alpha ketoglutarate via ALT to produce pyruvate. Amino group goes to urea cycle to be excreted. It's basically what I've just explained, okay, plus the help of this reaction, okay? Where were we? Okay. The transport of ammonia to liver. Now, sometimes the muscle doesn't need extra energy, okay? So you have another cycle that involves glutamine, okay? Now, glutamate, okay, becomes glutamine by adding another ammonia group, okay, through an enzyme called glutamine synthetase, okay, ATP and ammonia, okay, they join together with glutamate to give you glutamine. ATP is being used by the enzyme, okay, so the energy from ATP is being used for the enzyme to catalyze this reaction, giving me glutamine, okay. Glutamine, okay, keep in mind that glutamine has two ammonias in it, okay, two ammonias, two amino groups, okay. So, and glutamate has one. Glutamine becomes glutamate, and then glutamine becomes glutamate. Glutamine needs to release its ammonia, okay. For glutamine to become glutamate, it needs to release its ammonia. So we already one ammonia down from glutamine, which is glutamate. This is a bit confusing. Just take your time with the cycle. It'll be fine. The enzyme called is glutaminase, okay? I don't think that you need it for the exam, but just keep that in mind. Glutamate becomes alpha ketoglutarate through glutamate dehydrogenase, which is oxidative deamination, okay? And the ammonia is released. So now we have two ammonias here that are being released from glutamine. Okay, the alpha ketoglutarate, okay, with alanine becomes glutamate. So this is the glucose-alanine cycle. These are two separate cycles, okay? But the whole point is that we're just transporting the ammonia to the liver so that it can be excreted, okay? Alpha-ketoglutarate, alanine, joins together. Well, joins together, yeah. Joins together, giving me glutamate and pyruvate with the help of ALT. Pyruvate, okay, becomes glucose, and the glucose is used in the glucose-alanine cycle so that it can, be recycled and all of that. When I have the alanine becoming glutamate, and then the glutamate being deaminated, oxidatively deaminated, another ammonia is lost. So from this reaction, one ammonia is being lost, okay? Two ammonias are being lost, and from this reaction, one ammonia is being lost, because it's only one ammonia ion that's being transported in the glucose-alanine cycle, okay? Glutamate and ammonia via glutamine synthetase gives you glutamine. Glutamine is a non-toxic carrier of ammonia. It goes to the kidneys or liver mitochondria, so it becomes glutamate and ammonia. Glutamate becomes alpha-ketoglutarate, ammonia-urea cycle to be excreted. Basically what I just explained here. Now, you have a problem in your body where some of the proteins are misfolded, okay? Those proteins can form aggregates, okay? And we don't want that whatsoever. What the body does as like a preservance mechanism is that it breaks it down. It breaks it down with the help of ubiquitin and proteasome. Please, please, please make sure you're very comfortable with this concept, because it has been asked on multiple on multiple occasions in both the TVL and the past exams. Okay, so ubiquitin, okay, tags the defective proteins, okay, so that the proteasome can eat, sorry, this is eats, this is not seeds, eats the tagged protein, okay? Now, C-terminus, okay, of the ubiquitin attaches to the lysine residues, the amino group specifically, which is this. Like you can see over here that this is the ammonia, this is the E carbon, okay? It attaches here. C-terminus attaches, okay? And then that protein has multiple lysine amino acids, so multiple ubiquitins are tagging this protein, so it can go to the proteasome, okay, specifically complex 26, and eats the protein, breaks it down, liberating the amino acids per se, and in this reaction, ATP is needed. Now, the process occurs in the nucleus and cytoplasm. The protein gets degraded into chains that are 7 to 8 amino acids long, and the amino acids can be reused. Ubiquitin is spared, so it can be recycled. Pretty straightforward stuff, I believe, but here's the full explanation anyways. Now, cystinuria, okay, there is a defective transport channel that affects these amino acids. The abbreviation is called COL or COLA. I prefer COL. And cysteine, ornithine, arginine, and lysine are found in the urine, okay? And that's bad. That's bad. Why? Because it causes the formation of caliculi. Okay, caliculi are basically kidney stones, okay? The defective channel is usually in the proximal convoluted tubule, okay, in this part of the nephrotic system, okay, and it's an autosomal recessive trait. I don't think you need to know that it's autosomal recessive there. It's an autosomal recessive trait, but it's there for the sake of completion. It appears in urine since small intestines and kidneys have the same transporter, therefore COL appears in urine. Now, there are 3 types that you need to know. Type 1, if it's heterozygous, so that means they're just carriers, okay? It has normal urinary amino acid profiles, okay? But if it's homozygous, so the person 100% has cystinuria, choline and cysteine is elevated, okay? Cysteine is two cysteines, okay, joined together through a disulfide bond. Don't need to know this, but just keep this in mind. Type 2 and type 3 is hyperexcretion of cysteine and divasic amino acids, which is lysine, arginine, and histidine. Okay? Too much cysteine, kidney stones. That's pretty much it. The most likely case that they will give you is someone who has one or specifically cysteine in their urine profiles. I believe that's it for this for this part. Now, clinical heart, which is Hartnup disease, another clinical, sorry, is Hartnup disease. There is no tryptophan, okay, and it's due to a defect in a neutral amino acid transporter that's found in the intestinal mucosa and kidneys, okay? So there's nothing to pull in the tryptophan. So tryptophan is found in the urine, okay? No tryptophan means no niacin, which is vitamin B3, with pellagra-like symptoms. Now, pellagra is dermatitis, dementia, diarrhea, okay? And you can see in the pictures, they have like a dermatitis. Dermatitis is very similar to eczema, so you would see like eczema-like rashes on their skin, here, on their hands. This is the picture from your slide as well. Okay. How you differentiate mainly is because pellagra happens in adults due to alcoholism or malnutrition. Hartnup happens in children who are under a lot of stress. Okay. So Hartnup is caught early on. Pellagra is a disease that's later in life due to alcoholism. Now, amino acids, this is just what you need to know. Like as a quick summary, glutamate is a temporary nitrogen carrier / pool reservoir. Okay, glutamine is a non-toxic carrier of ammonia. Alanine, on the other hand, is also a non-toxic carrier of ammonia, but from the muscle. Okay, and this is used to also produce energy at the same time. Alanine, the glucose-alanine cycle. Now, these are some questions. Obviously, this is a pre-recorded session, so I'm just going to read it, pause for a second, and you guys answer. Which of the following can be produced by reverse oxidative deamination? Glutamine, glutamate, aspartate, alanine, glycine. The answer is glutamate. Okay, glutamate is the only amino acid that can undergo oxidative deamination. Okay, aspartate, no. Aspartate alanine, they use AST and alanine, okay, and it's not oxidative deamination, it's transamination, okay. Glutamine has glutamine dehydrogenase as an enzyme, and glycine, I don't know why they put glycine, but here we are. Now, another question. What happens in transporting ammonia and its non-toxic, what helps in transporting ammonia in its non-toxic form? Glycine, cysteine, alanine, and glutamate. The answer is both C and D, okay? Alanine and glutamate, okay, they both carry ammonia in its non-toxic form, so the body can tolerate it. Alanine specifically in the blood, glutamate is more of enclosed spaces, so think cells, okay? And if glutamine was an option, it would be correct as well. Next question is, which B vitamin is required to transfer an amino group from glutamate to alpha-ketoglutarate? Okay. The answer is C, pyridoxine. Okay, B6. You can go back to the reaction, and you can see that PLP is being used for transaminases. Okay. Finally, this is a clinical scenario. A 5-year-old boy has had episodic periods during which areas of his skin would develop a rash, which would spontaneously resolve in a week to 10 days. An astute pediatrician told the boy's parents to give him niacin the next time this occurred and a high-protein diet. And when they did, the rash resolved in a day or two. A likely disorder that this child has is which one of the following? Cystinuria. Highly unlikely, because this is not a urine problem, this is a skin problem. Hartnup disease, definitely. Myasthenia gravis is an autoimmune response against the receptors in the synaptic cleft. Alkaptonuria and jaundice. Jaundice, not really, because the patient didn't present with yellow eyes or yellow skin. Alkaptonuria, it's a disease in the breakdown of specific amino acids. It's not the correct choice. It's B, Hartnup disease. That's it for this session. I hope this was beneficial for you. If you have any questions, please don't be afraid to reach out, and please scan the QR code and give me your feedback. Thank you.